It's now my great pleasure to introduce our first speaker, Dr. Dheva Setiaputra. Dheva and I actually did our PhDs in the same program at the University of British Columbia. So for me, it's really fantastic to get to hear more about what he's done in his postdoc. But in his PhD, which was with Calvin Yip, David conducted a number of great studies using structural biology and biochemical approaches to characterize large protein complexes, including histone cell transferases, SAGA, and NUA4. And he was awarded the prestigious Marianne Hewer Award for the best PhD thesis from UBC Biochemistry and Molecular Biology Department. He then moved to Toronto for his postdoc with Daniel DeRocha, working on the 53BP1 DNA repair pathway. In this, he's had a series of fantastic papers and a recent preprint characterizing this pathway. And I'll note that throughout his career, he's received a number of awards, including the NSERC Alexander Graham Bell Graduate Scholarship, a CHR Postdoc Fellowship, and the Cancer Research Society Next Generation Scientist Scholarship and Project Grant, which provides 1 year postdoctoral support followed by a 2-year operating grant, kind of like the K-99 for those in the States. And Dheva is on the job market right now. And so I think any department would be very lucky to have him. For those of you on search committees, I recommend soliciting his application. And if you do receive it, give it a very good luck because you really should be hiring him. But so with that, let me finish by saying that I'm very excited to hear Dheva speak today on his work charting the 53BP1 RIF1 Shieldin pathway and its role in DNA double-strand break repair. Dheva, over to you.
Thank you very much, Ben, for that very kind introduction. So yeah, thank you again for the organizers for inviting me here today to share about a little bit of some unpublished, well, I guess a preprint research that I've done on how we use AlphaFold in particular to chart the 53BP1 RIF1 Shieldin pathway and try to understand exactly what the protein-protein interactions are mediating this pathway's role in DNA double-strand break repair. So everybody here knows that DNA double-strand breaks are extremely deleterious lesions, and they must be fixed to maintain genome stability. Some key sources of DNA double-strand breaks, especially in the context of cancer therapy, is that of ionizing radiation and radiotherapy, and also certain types of chemotherapeutics also induce double-strand breaks in the cell. So I'm going to oversimplify a lot about the DNA double-strand break repair pathways. I'm just going to tell you that there are two primary pathways that I'm going to talk about today for fixing double-strand breaks. And that is the first one being the very simple one of non-homologous end joining, where the ends are directly ligated together, and a more complex pathway called homologous recombination, where the break ends are resected to reveal long single-stranded DNA. And this single-stranded DNA is eventually bound by the RAD51 recombinase, which allows it to invade homologous templates for error-free repair of the break itself. Now, the choice between these two pathways takes into account many different factors, but I'm going to talk about two pathways that, or two proteins that mediate the choice between these two pathways, and those being BRCA1 and 53BP1. Just again, broadly oversimplifying, BRCA1 promotes homologous recombination, whereas 53BP1 promotes non-homologous end joining. What came to light is that these two proteins are mutually antagonistic in the sense that 53BP1 suppresses BRCA1 and vice versa. And this mutual antagonism really defines the proper timing and extent of these repair reactions so that they happen in the correct fashion. And the tension between these two pathways is very well illustrated when we talk about a certain type of cancers and the therapies associated with it. So many of you have probably heard of the BRCA1 gene, in that BRCA1 mutations results in the development of hereditary breast and ovarian cancer. Having mutations in BRCA1 dramatically increases the risk of developing these types of cancers due to the genome instability associated with loss of homologous recombination activity. But this loss of homologous recombination DNA repair activity also rendered these types of tumors very sensitive to certain types of chemotherapies, in particular poly-ADP ribose polymerase inhibitors or PARP inhibitors, and BRCA1 deficient cell or BRCA1 mutated cells that are homologous recombination deficient are extremely sensitive to PARP inhibition. and it is currently in the clinic to treat BRCA-mutated breast and ovarian cancers. What was very striking is that loss of 53BP1 in a BRCA1 deficient background actually rescues this PARP inhibitor sensitivity. And this happens because in the absence of BRCA1, 53BP1 suppresses homologous recombination such that it doesn't occur to the level required to repair PARP inhibitor associated lesions. But then when you lose both 53BP1 and BRCA1, you get de-repression of homologous recombination and you rescue the PARP inhibitor sensitivity of these cells. I apologize, I'm in a hospital, so sometimes there will be strange announcements. I've discussed DNA double-strand breaks as a pathological condition, but they're also physiological intermediates. And what I'm going to talk about today is that an immunoglobulin class switch recombination. So when B cells mature, sometimes they decide to switch the type of immunoglobulin that they express. And this is mediated through the induction of DNA double-strand breaks at two distal regions in the immunoglobulin locus, and long-range non-homologous end joining joins these two breaks together to recombine this gene such that a different type of immunoglobulin is expressed. And 53BP1 is especially required for this type of non-homologous end joining, more so than kind of broader bulk non-homologous end joining. So class switch recombination is a very characteristic function of the 53BP1 pathway. So I've spoken to you a lot about 53BP1, and as I mentioned, this is the topic of my study. What exactly is 53BP1? What is a very large protein with over 2,000 residues. Its N-terminal half consists of a disordered region that's highly phosphorylated by the DNA damage repair kinase ATM. And directly adjacent to that, it's a focus forming region, which contains an oligomerization domain and two post-translationally modified histone binding domains, that is a Tudor domain and a ubiquitin-dependent recruitment region. This region is called the focus forming region because of one very characteristic activity of 53BP1 is that it forms this very large foci around double-strand breaks when visualized by immunofluorescence microscopy. And each of these foci is thought to surround one or more breaks. So this is the Fragile Nucleosome seminar, so I'd like to kind of talk a little bit about how 53BP1 is recruited to sites of DNA damage. Well, very broadly speaking, so there is a constitutive dimethylation on histone H4 lysine 20 that is bound by the Tudor domain of 53BP1. And in the presence of a DNA double-strand break, this activates the ATM DNA damage kinase, which phosphorylates histone H2AX at serine 139. And this is the very famous gamma H2AX modification. And this phosphorylation mark is bound by the protein MDC1, which recruits a cascade of E3 ubiquitin ligases RNF8 and RNF168, which ubiquitinates histone H2A at lysine 15. And this ubiquitination mark is what is bound by the ubiquitin-dependent recruitment domain of this focus-forming region. So this is how 53BP1 gets rapidly recruited to sites of DNA double-strand breaks. Now I'm going to summarize kind of a pretty convoluted story in that 53BP1 is a recruitment scaffold that's responsible for bringing in downstream effectors, namely being RIF1 and the Shieldin complex that we and others discovered a few years ago. And the Shieldin complex consists of the following subunits here, and it is recruited by RIF1 to 53BP1 bound sites through RIF1's direct interaction with phosphorylation marks within its disordered N-terminus. Exactly how this pathway mediates DNA repair is still a subject of extreme interest, but there's some good, excellent work coming out of the Delanga lab showing that this pathway actually recruits the telomeric associated complex CST polymerase alpha to reverse DNA end resection by filling in the resected DNA. Essentially, this entire pathway then is what promotes non-homologous end joining and characteristic class switch recombination and suppresses homologous recombination. So we had a lot of data coming into this in terms of what this recruitment cascade looks like, but we had precious few kind of structural basis for these types of interactions. And we wanted to implement some new technologies to kind of systematically interrogate this pathway that we know a lot about to understand exactly what protein-protein interactions are within it and whether we can find new ones that help us explain how this pathway works in DNA double-strand break repair. And as I mentioned before, we implemented or we leveraged this new algorithm, AlphaFold multimer, to look for pairwise protein interactions that might be important for this pathway function. So we set out with these following questions. Can we recapitulate known interactions within this pathway and provide a structural basis for them? But can we also find novel binding interfaces here? And can we uncover that these new binding interfaces are biologically relevant? And I will mention that a lot of the work I'm going to present here is done by a talented undergraduate under my direct supervision, Shireen. And the work here is actually in the subject of a preprint that is available on bioRxiv. So essentially what I did was I took every single unique pair within this pathway that we are interested in and predicted a dimeric structure using AlphaFold multimer. We scored these heterodimeric structures for the confidence of predictions, and we took the highest confidence predictions and kind of evaluated them in comparison to the published literature, and also followed up on several of them. So this is a very busy figure, but it summarized a lot of the interactions that we're able to find within this pathway. And don't worry if it's a little bit small. I'm going to kind of go through a few of them with you now. The first two that I would like to show, it's just in comparison with some published structures that are already available. As you can see, AlphaFold multimer was able to recapitulate a lot of these multimeric structures to a pretty decent accuracy, at least at the level that we're interested in. Now I'm going to focus on one set of interactions, and that is between RIF1 and 53BP1. So AlphaFold pointed out that there is a disordered region of 53BP1 with its N terminus that is bound to the alpha helical repeat region of the RIF1 N terminus. And as you can see here, it orients two serines followed by two leucines that are directly kind of pointing towards RIF1, and the serines are pointing towards two basic residues upon RIF1. What's even more interesting is that for each pair of proteins, we predicted 5 different models, and within these 5 models, there's actually 2 different conformations of this binding. But both of these conformations uses 2 serines pointed to 2 of these basic residues and the 2 leucines that are directly preceding it. The reason why this was so interesting to us was because just the year before, we published a paper where we identified that 53BP1's phosphorylated N terminus binds RIF1 at these 3 distinct regions. And 2 of these 3 different motifs that we found were recapitulated in the AlphaFold predicted structure. So potentially we could have saved a couple of years of my life and identified them by AlphaFold, but it was nice to kind of get validated for that. But what's even more interesting, as I mentioned before, this interaction is mediated by phosphorylation marks. And we know this because if we take peptides corresponding to these regions, it requires phosphorylations to bind to purified RIF1. So what AlphaFold is able to do is it's able to infer the presence of these phosphorylation marks, but it's still able to detect this interaction. So, and this is kind of going back to last year's paper, but these 3 motifs are biologically relevant because if you disrupt them either by mutating one of the 3 leucines, or each of the 3 leucines, or all 6 of the phosphorylated residues, you get loss of RIF1 localization to sites of DNA double-strand breaks. And you also get loss of class switcher combination efficiency. And you also get loss of PARP inhibitor sensitivity as measured by aberrant chromosome formation in response to olaparib treatment in a BRCA1 deficient setting. And what's also really nice about the AlphaFold prediction is that it really gave us a context of why these leucines are very important. As you can see, the leucines are buried in the hydrophobic groove of RIF1. So it was really nice to get some sort of a structural basis for these observations that we found previously using a completely different set of methods. Now that's recapitulating an interaction that we found. Were we also able to find a new interaction? And that seemed to be the case. What AlphaFold predicted was that the RIF1 heat repeats binds to the C terminus of shield 3. And I will remind you that we have previously determined that shield 3 is the most upstream subunit of the Shieldin complex, and that RIF1 is required for Shieldin recruitment, but we didn't know exactly what domains mediated this and whether or not there's any other proteins involved. What AlphaFold suggested was that's not the case. So we tried to validate this completely novel prediction, and we did so by expressing these fragments in insect cell systems and did some pull down analysis. I'm just going to direct your attention to these regions here. You can see with full length shield 3, you're able to pull down both RIF1 and REV7. With the N terminus, you're only able to pull down REV7, and this is already known. And with the C terminus, you're able to pull down RIF1, very consistent with the AlphaFold predictions. Of course, we wanted to test in cellular assays whether or not this interaction was important. So we turned to this FOC1 lac repressor assay where we induced DNA double-strand breaks at lac operator arrays and tested for recruitment of GFP tagged prey proteins to these double-strand breaks. You get this really nice focus where the FOC1 is recruited to sites of lac operators, where it induces DNA double-strand breaks. As you can see with full-length shield 3, you get nice localization to this focus, but then with only the C terminus, that is the only fragment that's able to mediate this localization. And this is a quantification here. And we did a lot of other analysis, but we also zoomed into the interface between RIF1 and shield 3, and we identified individual residues that are likely important for this interaction. When we mutated them, we were also able to see that some of them completely abolished the recruitment of shield 3 to sites of DNA damage. So even to a single residue level, it looks like the predictions we made were, to a high degree, quite accurate. Of course, is this biologically relevant? Again, this time we wanted to see whether this interface is important using class recombination assays. This time we use a mouse B-cell lymphoma system that switches from IgM to IgA when stimulated with this cocktail. And I kind of remind you here which mutations results in abolishment of the shield 3 RIF1 interaction. And as you can see, when you have these point mutations, you get complete loss of rescue of class switch recombination activity in this class switch recombination assay in shield 3 deficient cell lines. Whereas in the point mutations that are able to retain some of this interaction, you actually are able to complement it back to wild type levels. So that brings us to the end. So we set out with the following question: were we able to recapitulate known interactions and provide a structural basis? And yes, especially with the one I spoke about with the phosphorylated 53BP1 RIF1. There's actually multiple others that really very pleasantly surprised us that the analysis was able to turn up. Were we able to identify a novel binding interface? And that did seem to be the case of the shield 3 C terminus and RIF1. And whether or not these interactions are relevant, yes, certainly that what AlphaFold predicted is the true binding interface that's required for cells to mediate this DNA repair pathway. And so that brings us to the end of my presentation. Of course, I'd like to thank the other members of the Desrosiers Lab and the organizers for inviting me, and I would love to answer any questions that you might have.
Awesome. Thank you, Dheva. That was really cool. That was a fantastic talk. I was wondering, I'm fascinated by this approach of using AlphaFold to predict protein interactions as like the starting point to then go and test either in vitro or in cells. How much, like, how general do you think this approach will be? Like, will we start seeing this be widespread, or is it like needs a lot of work to get it to work?
So I think it's actually quite easy to get it to work. It's very fast. All you need is sequence information. It's just that I think the field is still working on the best ways to weed out false positives, right? Because those are the dangerous parts. The nice thing with doing it on a system or in a pathway that we know a lot about is that we have a lot of intuition and knowledge and experimental evidence that supports any single kind of prediction. So we were able to kind of use that to filter out a lot of these false positives. So yeah, I mean, already high throughput. I'm pretty sure there's going to be a network of every single protein to every single protein already in the works right now, but you really have to be careful with it. But it is very powerful and there's no reason not to use that.
Yeah, and based on what you've done, Like, are you starting to get a sense of like what sorts of false positives come out of this?
It's actually very interesting. There seems to be kind of like a nodal effect. Like there are certain proteins that are just beacons for false positivity. And I'm not exactly sure why that is. It might just be the type of the fold that it has. But it's certainly something interesting that has to be taken into account.
Will this be similar to like what they call it, the crapome, like everything that comes out of an IP mass spec?
I think that's actually like physically that, that's based on
Yeah.
physics, whereas this is a
Yeah.
little bit more about the training. So it might not be the
Yeah.
same. Yeah.
Yeah. Awesome. Thank you. If anyone else have any questions, you can post them in the Q&A or raise your hand. I was wondering about the PTMs. Like I was really interested that you could with the RIF1 interaction or the 53BP1, like you could see the interaction, even though in like when you do the experiments, you need the phosphorylation there, but obviously that's not there. So like, how do you think it's able to be tolerant of whether the phospho is there or not?
Yeah, so you have to, so AlphaFold was trained on the PDB, right? And the PDB has lots of structures that has post-translational modifications. It just wasn't coded into like the actual output of AlphaFold itself. But the training, I guess, just looks at the environment of the structure around it and kind of infers its presence. And this has been shown before, which is very interesting.
Yeah, that's fascinating. I see Felix, you've got your hand raised. You want to ask your question?
Cool. Thanks. Very nice talk. I was wondering, you actually detected an interaction between an intrinsically disordered region and the folded domain. Is this intrinsically disordered regions kind of short, so they usually are in crystals, or is it like a really long intrinsically disordered region where where you could detect these interactions?
Yeah, so these ones are actually very long. The 53BP1 disordered region is like, I think like 1,000 residues long or something like that. And actually in the DNA repair field, or at least the proteins that I've looked at, it often has these long disordered regions. And somehow in my experience, AlphaFold is able to predict these interactions with folded domains with relatively high accuracy.
Oh, that's nice. Looking forward to try it myself.
Yeah.
Thanks for showing it.
Yeah, thank you.
Awesome. Thank you, Dheva. We'll wrap it up there. If you have further questions, you can post them in the Q&A or just get in touch with Dheva yourself. Like I said, he's on the job market, so also solicit him to apply to your postings.
Thanks.
Thanks again for the great seminar.